explores the adaptation of the native yeast system into Drosophila to investigate gene-targeted expression. This system, known as the Gal4 UAS system, expands the capabilities of Drosophila as a model organism. Through driver and responder assignments to male and female flies, the resulting progeny can express any number of targeted genes to allow the researcher to view phenotypic effects and mutations on development. I will specifically be talking about Figure 7 in this article, but in order to do so, information prior to it has to be described. The basic Gal4 UAS system has been adapted from yeast and is actually quite clever and works as such. Gal10 and Gal1 are genes located on a chromosome that have adapted enhancer and inhibitor characteristics. 17 base pairs between the locus of Gal10 and Gal1 lies in upstream activator sequence, or UAS. The name is self-explanatory. When something is bound to the site, transcription of Gal10 and Gal1 is enhanced. In this case, Gal4 is that thing. When the Gal4 protein is bound to the UAS, specifically in the presence of galactose, Gal10 and Gal1 transcription is enhanced. The inhibition of this system is in the absence of galactose. When galactose is not present, another protein whose gene is located somewhere else on the chromosome, known as Gal80, binds to Gal4 and blocks transcription of Gal1 and Gal10. This is the basic mechanism behind the Gal4 UAS system in yeast. This system has now been adapted to work in Drosophila in a slightly different manner. The first Gal4 UAS system adapted to work is as such. A driver fly, which I will arbitrarily pick as a male for this model, carries the Gal4 gene. This gene encodes for a protein, but the protein has no function in the male because there is no UAS for it to bind to. Now we have a female fly who is called the responder. The responder carries the UAS site next to a specific gene. I will use GFP in this example, a green fluorescence tag. Even though the female carries the gene of interest, often lethal, she will not die because she does not have Gal4 to enhance transcription. When the male and female mate, the recombinant progeny will have both the Gal4 and UAS site, and so they will glow green, because now the Gal4 protein can bind to the UAS site and promote transcription of GFP. Only the progeny who are recombinant will express GFP because they had to receive the Gal4 aspect from dad and the UAS aspect from mom. So not all progeny will express GFP. This is the basic Gal4 UAS system, which has been adapted to promote transcription of genes in progeny, often human genes, so their effects and mechanisms can be understood. This paper continues to speak about recent modifications of the Gal4 UAS system to make it easier to control, more specific, and more inducible. The modification I will be highlighting is the adaptation of the Gal80 inhibitor in the FLP-FRT system from yeast to make the Gal4 UAS have increased inducibility. I have not mentioned the FLP-FRT system. This system has also been adopted from yeast and works as such. FLP is a recombinase that promotes recombination between two FRT sites. In order to transcribe a gene, there has to be a promoter in front of it. That's basic Bio14. To block transcription of Gal4, an FLP out cassette, which consists of two FRT sites, is placed in between the promoter and the Gal4 gene to block transcription. FLP is induced by heat shock, and when it is induced, the FLP out cassette is removed, and the promoter leading Gal4 is turned on and the system is functional. When all of these methods are combined, an incredibly inducible system is made. Through recombinations that the cells already bear, cells will become Gal80- using the FLP-FRT system and therefore express Gal4 continuously. In cells that are heterozygous, Gal80 blocks transcription of Gal4, and then through FLP-induced recombination, all daughter cells that are homozygous for the mutation of interest will no longer have Gal80 and can therefore express Gal4 and in turn GFP. One point that this paper raises is that there are not many responder constructs commercially available for use. This can make it difficult to study specific genes and make the process much longer since the constructs have to be made from scratch by the researcher. The advances of the Gal4 UAS system will expand the use of Drosophila and allow the study of human genes easier in vitro. Many adaptations have been made through hormone reception and other inducers to make the system easier to use. Even though our lab will only use the basic Gal4 UAS system, being able to comprehend its advances will make a better understanding of the original.